Alright, welcome everyone to part one of our new series of lectures on lipids. And today's going to be on essentially generally what are the different lipid types and how are lipids metabolized. So first thing you want to know is that glycerides, which are a major part of the structures in our body, are comprised of a glycerol backbone and at least one fatty acid. So that's a glyceride. We have monoglycerides, diglycerides, triglycerides. Now, if you take a fatty acid itself, if you look at the top fatty acid here, this is a saturated fatty acid, and basically what you see is that the carbon atoms have their full quota of hydrogen bonds, or hydrogen atoms with no double bonds. A monounsaturated, however, is the second fatty acid that you see, and that has one double bond. Whereas if you have more than one double bond, it's going to be a polyunsaturated fatty acid. We can sort of differentiate lipid types into simple, compound, and derived. So if you look at a simple, which would be sort of like a free fatty acid, that's just glycerol or a glyceride. It's glycerol plus a fatty acid plus fatty acids. Compound is essentially simple bound to other substances like phosphate groups. So a phospholipid, which makes up a lot of our membranes, for example. And then we have derived, which are simple and compound lipids combined. That'd be like cholesterol, hormones, fat-soluble vitamins. One thing to realize is that one thing they say is, oh, you are what you eat. Well, one thing that's important to emphasize is that the level of saturation of a fat will in fact impact things like membrane fluidity. So this is essentially an unsaturated fatty acid here, and if you look at the membrane here, one thing you notice that essentially the unsaturated hydrocarbon tails of phospholipids have kinks that keep molecules from packing together. That enhances membrane fluidity. In contrast, if you look here at saturated, you're able to keep the molecules packing close together, which makes it more viscous and not as fluid. So one thing we know is that if you have that the fluidity of your membrane is going to be important for things like transport of various nutrients, insulin sensitivity, etc. We can also define fatty acids by their structure. In general, they're about 12 to 24 carbons in length. However, we can differentiate these into short, which is less than 6 carbons, medium, which is 6 to 10 carbons, and long, which is greater than or equal to 12 carbon atoms. And then the nomenclature of fatty acids, basically a lot of times you'll see things like 18,3 omega-3, for example, if you look at alpha-linolenic acid. Okay, each of these numbers has a specific meaning. So the first number, which is here, you can see 18. That's the carbon chain length. The second number is how many double bonds that there are. So in this case, there's 3 double bonds, which makes it a polyunsaturated fatty acid. The third number tells you where the double bond starts. That starts on the third and fourth carbon, which you can see. So again, first carbon is going to be the carbon chain length. Second carbon is going to be the number of carbon double bonds. Third is going to be the first position where the double bond occurs. Humans can't interchange between omega-3 and omega-6 fatty acids, and they also can't synthesize fatty acids with a double bond until the 9th carbon. That makes omega-3 fatty acids and omega-6 essential, meaning you have to obtain them through diet. Here we're looking at metabolic fates of omega-3s or N3s and N6s or omega-6s. If you look over to the left at the omega-3s, high in things like flax, obviously high in fish. One thing you notice is that they are anti-inflammatory. However, if you look at omega-6 fatty acids, they end up being pro-inflammatory. So that's very, very important to understand. So, one of the things to understand is that the essential fatty acids essentially operate through things called eicosanoids. These are biologically active substances like prostaglandins and leukotrienes that are produced by the direct enzymatic oxidation of polyunsaturated fatty acids. They're produced in most cells, and there's over 100 eicosanoids, docosanoids, and autocoids have been identified that are linked to these omega fatty acids. Omega-3 fatty acids, the primary fatty acids that get the most attention is EPA and DHA, and notice that they regulate gene expression. They also have derived substances like eicosanoids, which have signaling effects, and they also help with structure, such as the cell membrane. So here's one thing I want you to know. If you look at long-chain fatty acids, which is here, one thing I want you to notice here is that long-chain fatty acids are first taken up into the small intestine. They then have to go through the smooth endoplasmic reticulum where they're converted into triglycerides. Then they go through the then triglycerides plus cholesterol and protein goes through the Golgi tendon apparatus, which goes to chylomicrons. That goes in lymphatic vessels and eventually dumps back through the jugular into normal blood, and then it can get used as fuel. In contrast, if we look at our medium chain, diffuses straight into the enterocyte, directly into the blood. So that's very important because basically medium chains don't need a transporter. Long chain fatty acids have a lot more complex mechanism of action. These are just some common fatty acids that we could see. If you know, the two of the main essential fatty acids discussed are linoleic acid as well as linolenic acid. Okay. We also have arachidonic acid, which our lab's done a lot of research on. Palmitic acid is very common. So here's the thing. One of the things I realize is that if we look at adipose tissue and how it's metabolized, adipose tissue is a complex endocrine organ. It doesn't just store triglycerides or triacylglycerols. It also has endocrine functions that regulate metabolism, insulin sensitivity, and systemic inflammation. So, how does lipolysis occur, or the liberation of fatty acids from the adipose tissue? Well, this seems to be regulated by hormone-sensitive lipase. And hormone-sensitive lipase catalyzes fatty acid cleavage from its glycerol backbone. It's activated by phosphorylation under conditions of low insulin and the presence of epinephrine and norepinephrine. So you can see that here. You see epinephrine and glucagon essentially stimulate hormone-sensitive lipase, which cleaves off fatty acids from the triglyceride, and then those enter into the into beta oxidation, where basically the fatty acids convert it into acetyl-CoA, and of course that can enter into the citric acid cycle, and then that will go to the electron transport chain. Insulin and citrate inhibit this response. So high insulin, low epinephrine, impaired ability to store, or impaired ability to take in and oxidize fatty acids. Low insulin, high epinephrine and glucagon, the opposite. Liberation of fatty acids occurs and their oxidation is heightened. So our muscle can take up fatty acids from either the plasma as free non-esterified fatty acids that have been liberated already, or it could be fatty acids carried as triacylglycerol and lipoproteins. In this case, you need lipoprotein lipase to cleave off those fatty acids. One of the major things that you want to know is that blood flow is a major determinant to the extent of fatty acid uptake and oxidation. Everyone's heard of ketone bodies, or most of you I'm sure have. One thing that one of my old professors used to tell me is that carbohydrates burn in the flame of fats. So when carbohydrates are low, or fats burn in the flame of carbohydrate. So when carbohydrates are low, acetyl-CoA can't readily enter the mitochondria. So it's converted to acetoacetate and hydroxybutyrate, D3-hydroxybutyrate, which are ketone bodies. And organs can use ketones instead of glucose, including the brain, particularly when carbohydrate is low or during starvation. So when ketones are high, we know that our primary source of fuel is fat, and we switch over from carbs. Now, fatty acids can be synthesized de novo, meaning in the body, from other sources like carbohydrate. Essentially, you can have acetyl-CoA as a starting molecule, and you add 2 carbons at a time. If you add 2 carbons at a time until you get to 16, you would have palmitic acid, which is the most common form following fatty acid synthesis. So in summary, fats can be simple, compound, or derived. Fats can be saturated or unsaturated. And long-chain fatty acids require a transporter for oxidation, while medium and short do not. That could have certain implications on sport performance, as we'll discuss later. Essential fatty acids must be obtained through diet and exert their effects through eicosanoids. Fatty acids in the blood must be cleaved off via lipoprotein lipase, and in the tissue via hormone-sensitive lipase. And lipolysis is regulated by catecholamines and glucagon, as well as insulin. Catecholamines are high, glucagon's high, and insulin's low. Fatty acid metabolism is maximized. That's the end of Lecture 1. I'll see you for the next lecture.